Hello everyone, I'm Roop Nandini and my project is Speckle noise reduction in OCT images. This page contains all the contents of the project report and it includes introduction to what OCT images are and why do we need to reduce the noises in them. Then what are the tools and packages that have been used, how is dataset defined, estimation of jitter, what is jitter and its removal process. Then how we have denoised the noise, and then supervised learning approaches, and finally we conclude. So OCT is an acronym for optical coherence tomography and it provides high resolution cross-sectional images of biological tissues. However, these images are affected by speckle noise and it degrades image quality and obscures fine structural details which are significant for accurate diagnosis and automated analysis of retinal layers. So in this project, that is the challenge that we address. So we try to reduce speckle noise in OCT images through classical image processing techniques, which are combined with deep learning approaches. So the basic framework is to understand the data set, then understand what jitter is, understanding how to remove it, then uh, the denoising methods and supervised learning using unit architecture. The ultimate goal is to enhance image quality while preserving critical anatomical information necessary for retinal layers, segmentation automatically, and automatic clinical analysis. Then I have discussed about what OCT images are and how they are represented. So they are represented either as a 2D cross section or a grid or as a 3D volumetric data. If the 3D volumetric data is taken over a period of time, then time can be considered as the fourth dimension. So it can also be considered as 4D data. So what does each pixel or every grid point expresses? Each pixel intensity corresponds to the backscattering properties of the tissue at that location. So the brighter regions indicate higher reflectivity and it allows clinicians to identify different anatomical structures and detect the abnormalities. So because of speckle noise, image quality is reduced and overall image clarity and structural details are lost. There are segmentation difficulties between different layers of retina, which leads to diagnostic uncertainty, which obscures clinicians to understand the pattern or can lead to false positives. Um so then we have talked about retinal layer segmentation. So there are 8 different retinal layers. The first one is inner limiting membrane. It is the innermost boundary of retina which separates the retinal nerve fiber from vitreous humor. It is the first reference boundary. Then comes the nerve fiber layer. It contains the first part, that is the axon part of the, if the neuron is like this, then it contains the head part of the neuron, which is the axon. Uh, if this layer becomes thin, it is an indicator of glaucoma and other optic neuropathies. Then the ganglion cell layer uh, comprises of the cell bodies of the nerve cells, and if there is any change in thickness in this layer, so changes in thickness in this layer is associated with retinal and neurological conditions. In on this page, the largest is in a plexiform layer. It contains synaptic connections between bipolar cells and ganglion cells. Uh, so this plays role in signal processing within the retina when the signal from eye has to be delivered to brain. This is the place that is crucial. Then we have inner nuclear layer. Alterations in this can indicate inflammatory or degenerative conditions. And then we have 3 more layers, that is outer plexiform, outer nuclear, and retinal pigment epithelium. So then we have talked about the tools and packages that have been used in the project. So the core programming and environments environment that I have used is Python 3 as the coding language, and I have used Jupyter Notebook. NumPy is used as the fundamental library to compute and use the array operations. Pandas for data manipulation and analysis, and particularly for the Excel files that will be discussed further what they contain. So the image processing, for image processing and computer vision part, TIFF file is used. That is a specialized library that deals with reading multi-frame TIFF image stacks. So after that, I have used scikit-image um, and SciPy, and deep learning frameworks that have been used are PyTorch, Torch.nn, Torch.optim, and Torch.utils.data. For visualization and analysis of data, or what we are doing in the project, I've used Matplotlib. So it is important to validate what we are doing. So for that, we are using scikit-learn to split the data as training and testing data. So dataset exploration involves analysis of the image stack structure, annotation files, and their organization. So the primary dataset is the volumetric OCT scans that have been stored as multi-frame TIFF files, and alongside that, hand-annotated retinal layer boundary coordinates were also stored in Excel format. So so each OCT volume is stored as a stack of 2D B scans in TIFF format, uh, and Excel files denote the coordinates of the layers that were hand annotated by expert. So in TIFF image stack structure, the main file intensity.tif contains the raw OCT intensity values that represent back scattered light from retinal tissue layers. Each frame corresponds to a single B scan, that is a cross-sectional slice at a specific spatial location. Uh, After analyzing the data, the file format was TIFF, and I've discussed the data types, stack dimensions, intensity ranges. In addition to TIFF files, a MATLAB data file was also provided, which discovers or contains supplementary metadata or processed representations of imaging data. I analyzed this file using SciPy's MATLAB file reader to understand what its structure was and what was its relation to the raw TIFF data. Um, so then we come to hand annotated coordinate data. So retinal layer boundaries are annotated by hand by experts, and these coordinates of layers are represented as x and y coordinates, and in an Excel file. One Excel file contains x coordinates and the other y coordinates of a particular image, and they are paired together. So each row represents a distinct retinal layer boundary, and columns contain sequential coordinate values tracing the boundary from left to right across the image width. So variable length rows accommodate boundaries that may not span the entire image width, and empty cells indicate that it is the end of a boundary trace. So in hand annotated scans, we have 252 B scans. B scans is just a medical name for the scans that we are using in this project. So 252 scans have all the 8 layers that have been traced, and the annotation density talks about how many layers we are denoting. So when we see, so this is the graph that shows how many scans have how many layers. So apart from these, although we started with 339 scans, a lot of them were not even annotated, and about 252, exactly 252, sorry, were annotated with 8 layers. Some were with 4 and some were with 2. So to verify that the coordinates that we had for layers were actually overlapping with the images, we have used one image and the coordinates of the layers to verify. So the red layers are the annotation coordinates, and as we can see in this image that they do overlap with different retina layers, that is the scan in black and white. So we realized that the traces are quite comprehensive and cover layers completely, but we can also realize that there is some speckle noise impact. And so what this allows us to understand is that we need to validate the correspondence between annotations and image structure and identify the regions where speckle severely degrades image quality and layer visibility, and to demonstrate the completeness and quality of the actual true data, to provide with qualitative assessment uh, that if the denoised images are actually preserving anatomic descriptions. Um, so now we move on to estimation of jitter in OCT images. Jitter are basically small line movements that cause misalignment between consecutive B scan frames. And now we need to understand what jitter in OCT scans will do and how we need to correct it. So it refers to small random displacements between consecutive frames, and unlike systematic drift or large cyclic movement, it is rather of pixel or very small movement. So just only a few pixels in both horizontal or vertical direction. And this movement is comparable to the scanning rate or the frame acquisition rate. So when we characterize jitter on the basis of magnitude, direction, temporal pattern, and what its impact is when we average it out. So we further will move on to image registration algorithms, and it's important to understand jitter before that. And it provides quality metrics for the acquired volume. So, and that is what makes a scan good or bad. So, sources of jitter can be ocular tremor, blinking, or microseconds that are small and voluntary eye movement that occur during attempted fixation. Then we other reasons are drift, slow eye movements that accumulate over the duration of the time that we are taking the scans in, and cardiac and respiratory motion can also be responsible for these. Other than that, technical sources can be scanner instabilities, trigger timing variations, and subjectivity device coupling. So now we will move on to jitter estimation methodology. So to estimate jitter, we select one reference frame that is at the half of the sliced window, that is at S by 2 slice index, where S is the total number of slices, and then each frame goes under normalization and windowing. We used hand windowing in this, and it improves registration robustness. So the third step in jitter estimation process is a phase cross correlation uh, when we calculate the shift from the reference frame using phase correlation method that is available in Scikit image library. And the fourth is error estimation, and the fifth is shift validation. We validate if there are extreme shifts, and we can consider them as outliers, or we need to consider that as jitter. Uh, so the implementation parameters are further discussed, and then results and visualizations are also shown. The left image in this shows shift in pixels with respect to x and y coordinates, and the right one shows the normalized registration error for each slice, indicating alignment quality. And we realize that the error for per slice, the normalized error per slice is quite low, that is lower than 0.2. So uh, um, and the median absolute vertical shift and horizontal shift are also under 50 pixels. Maximum observed shifts were although more than 95 pixels in both dimensions. Slices which have higher error, that is more than 0.2, sorry, may indicate degraded image quality or extreme motion or say blinking. So now we know what kind of jitter our images have, and we need to remove it. We need to remove it because we want to create pseudo-clean images which can be used as target or reference images when we train our models. Uh, so the jitter removal process contains different parts. First one of which is image registration technique. So what we have used is phase correlation registration, which is a frequency domain technique that offers several advantages for OCT image alignment. Why this was necessary? Because it is robust to intensity variations. It means that it is insensitive to linear brightness changes and contrast variations, which makes it suitable for OCT images where intensity may fluctuate due to signal strength variations, and intensity is not related to image quality. So then I have defined the mathematical formulation for the same, and it for the pre-processing for registration contains three parts, that is mean centering. So the mean of so the image of so the mean of image is removed, that is the DC component. That mean is basically the DC component, and if we separate all the it from all the images, then only the differences will be available. Then we use standard deviation normalization and hand windowing. Hand windowing is a method to hand win- is a method to preserve edge and boundary points. So how it's done is that we take one image and we compare it with around nine of its neighboring images, like four back and four forward, and we use all those nine images to create one better image. Uh, so we give our model, we give code to use from 3 to 9 images uh, to create one truer, better image. So usually we have used 9 images, and then I've also talked about the mathematics behind it. Then what we actually do is create confidence maps that through which we can generate uh, confidence maps, and it shows how we are improving images and what is happening. So the formula for confidence interval is given, and also the implementation parameters and keywords are defined. So this is the reference image and this is the pseudo clean image. We can see that the vertical lines are not as jittered. So the jittering has decreased in the pseudo clean image. This is and the confidence map shows that the yellow part is where it is completely confident, and this part also it has turned somehow yellowish, and the bottom part also is yellowish. So the edges are going to be blue because uh, that is where the shift happens, but overall the quality and the difference between the images show that there is some improvement in the new jitter-free pseudo-clean image. This is another example of the same thing. So the average number of frames that were used to create one pseudo-clean images was 6.8. It could have used anything from 3 to 9, so average was 6.8. For about 15 to 20% of the images were outliers and they have been removed. So the confidence distribution in the tissue region is about 0.75 to 0.85, which is good, and Also, another thing about this is that we cannot try to make the image better and better, because then we might lose some boundary points, and important information can be smoothed out, which was necessary. So we cannot just make the image better uh, de- in mathematical terms or with numbers, but we have to understand what is good and what is workable with. So I've also mentioned some limitations and residual artifacts about this process. And then we come on to baseline denoising methods, which we'll cover after. So median filtering is a non-linear and order statistic filter that replaces each pixel with median value within a local neighborhood window. And uh, unlike linear filters, it is particularly effective at removing impulse noise while it preserves the edges. So for preliminary image enhancement, it is often used. N is a square neighborhood window, and it is centered at the particular pixel that we want to that we are focusing upon. And in this, a 3 cross 3 window is used, which provides a balance between noise reduction and computational efficiency. So through the neighbors are used to calculate the value of that particular pixel using the medians. It has several advantages, that is that it preserves the edges, it is computationally effective, and there is some definite deterministic output. The limitations are that the noise reduction is limited, because we cannot use uh, larger window or very small windows. Very small windows, although are better at preserving edges, but those are way more computationally ineffective. And if we use very large windows, there is texture loss. We know that there is, although granular uh, noise, but there is also finer details that can be lost if the texture is lost. It is also not very effective in treating Gaussian-like noise. So in median filtering, we were only using the nearest neighbors, the exact neighbors of of a pixel cell. But in non-local means, it does not use the exact neighbors, but similar patches that are found across the image. Um, so it is well suited to medical images where anatomical patterns are repeated across spatial locations. It does not focus on the coordinates that are exactly near to what we are focusing upon, but the overall uh, image. So the method description has been shown. Um, so this method is better than median filtering, as it is just not averaging out the local neighbors, but is focusing upon the image as a whole. And now we will look at the comparative quantitative analysis part. We are using comparative analysis to understand the difference between two images and if we are actually improving. So there are two evaluation metrics that we are using. First is PSNR, that is peak signal to noise ratio, and the second is structural similarity index. Uh, the larger the PSNR, the better the signal strength is, and the second metric, that is SSIM, lies between -1 to 1. -1 indicates that there are no similarities, and 1 indicates a perfect similarity. So the two methods that we used were median filtering and non-local means. So the uh, raw image has a PSNR, or the average PSNR, of 23.080 and SSIM of 0.515. And then both median filter and non-local means, both PSNR and SSIM have increased. So we realized that there is consistent improvement. If only one if only one of the metrics was increasing, or if only one of the methods the metrics were increasing, then we could not have been sure if our method is actually working. But the consistent improvement shows that both methods are improving the image qualities. NLM is superior because it increases PSNR and SSIM both more than median filtering that we have already discussed, because it is focusing on the image as a whole. So this is just a visual comparison of the raw image, the pseudo clean image, and the image that is produced by median filtering and by known local means. So these are the two reference frames that we have used, and this is just for visual comparison. Although we see that there is improvement in PSNR and SSIM, there is also some kind of limitations that we are facing, that there is still incomplete noise removal and the boundary artifacts are not yet completely preserved, and over smoothing can lead to losing information. So we need to consider and answer these limitations. So what we are using now is supervised learning approach. We have already created target pseudo clean images that are our reference from as to what our image should be and what our image actually is right now. So supervised learning is basically when we know what we should have had and our prediction. So and we keep improving our predictions on the basis of the reference that we are using. When we have the reference, call it we call the learning approach to be supervised. When we do not have uh, any result or any reference to look at, then the learning approach is known as unsupervised. And third type is reinforcement learning, when we keep making mistakes and learning. And so basically we are using a supervised learning approach in this. So ICAP clean is what we predict. I clean is the clean target image that we are using, and we are training our model in the difference of these two things, I cap clean and I clean. So now we have split our data into train and validation data set. We are using 15% of the data set to be used as validation set and 85% to be used as training set. So the hyperparameters are tuned on the basis of validation performance, and uh, so now we have split the data into 221 samples are used for training and 15% are used for validation. Um, so the architecture that we have used is the unit architecture, which was originally developed for biomedical image segmentation. And it is highly effective for image to image translation and tasks like denoising. And it has a symmetric encoder and decoder structure with skip connections that enables global context generation and fine spatial detail preservation. So basically, it the left part or the blue part is the encoder part, and the green part is the decoder part. With So the encoder progressively reduces spatial resolution to encalcate and capture the hierarchical representations. So in level 1, we saw that the input was 1 cross H cross W. The image was a 2D image, and there was no component in breadth. So we have considered only height and width. So it converts it into a convolution block that is for 16 cross H cross W, and now the H and W are going to be decreased subsequently in each step. So in level 2, the dimensions are 32 cross H by 2 cross W by 2, and in level 3 it is 64 cross H by 4 cross W by 4. And it is the contracting part. H and W are decreasing, so it is the contracting part, and the decoder is the expanding part. And it is also known as the upsampling, and encoder is known as downsampling. So in level 3 of decoder, it is 32 cross H by 2 cross W by 2, which converts to 16 cross H cross W, and the output is a 1 cross H cross W denoised image. Some design choices that we had to make were group normalization instead of batch normalization, uh, because that to ensure stable training and due to the memory constraints of the laptop, uh, only group normalization could be done, and we have created a channel of 16 rather than 64 that is used in the original unit, as it reduces memory footprint and again a constraint of computation on our end, and it also reduces the training time while maintaining the representational capacity for the task that we have at hand. And we have used three-level hierarchy that more or less captures all that we wanted to. So we saw that at the same dimensions, there were some connections between the encoder and decoder parts. It is called skip connections, and it enables the network to preserve fine spatial details, that is, the edges and the texture, while the bottleneck learns high-level denoising strategies. So now we move on to training configuration and hyperparameters. The loss function for this contains the total loss consists of loss in intensity plus lambda times loss of edges. So the loss in intensity is mean absolute error between predicted and target images. It takes care of overall brightness and contrast fidelity, and loss in edge is mean absolute error computed on Sobel gradient magnitudes. Uh, Lambda is 0.35 that we have chosen to encourage edge preservations without overwhelming the primary intensity reconstruction objective. This was the best that value that was coming for this purpose. So the mean absolute error, which is also known as the L1 loss, is preferred over the mean squared error, that is the L2 loss, as it is more robust to outliers. In when we use L2 loss, which is also lasso regression 2 loss, it uh, it still contains some kind of outliers, and the optimization algorithm that has been used is the Adam optimizer. So we have um, used 20 complete passes through the training set, and after each epoch, we have used the validation set to uh, look at if the model is learning or not. We have looked at the PSNR and SSIM evaluation metrics after each epoch on the validation set. Um, so the checkpoint with the lowest validation loss is saved as the final model, and so we will realize that after the 20th epoch, the loss was increasing. So it is a subtle indication that we should now stop we should now stop the passes. So the training loss has decreased monotonically from 0.1096 to 0.76074. Also, the validation loss has also decreased. And after at the 19th to 20th epoch, we realized that the validation loss has increased, which is an indication that we should now stop with our uh, epochs. So we see that although there was a rapid initial improvement, it has now settled down to a uh, minimum value. After that, there will be only increase in validation loss, which is what we do not want. The PSNR has also studied. PSNR and SSIM have also steadily increased. So the consistent improvement in SSIM and PSNR show that um, there is improvement in the learning part, and the final performance is taken at the 20th epoch. The overfitting analysis is overfitting means that when our model, instead of learning the patterns of how to predict the output, it actually learns the output with reference to the input. We do not want overfitting. That is what we have discussed in this part, and so the consistent metric improvement shows that throughout training demonstrates that the model has not overfit, and the learning has been generalizable in denoising patterns. Uh, so the final model performance evaluation is as follows. Um, so this was the or raw noise input. We have compared it with the median filter denoising method, NLM method, and the unit that is a supervised learning technique that we are using. Then uh, we are going to look at the improvement versus raw and improvement versus NLM. So there is improvement in both the PSNR and SSIM with respect to the row and also with respect to the NLM. So the one point to mention in this slide would be that the results are computed exclusively on data that was not seen during training, and it provides an unbiased performance estimation that is applicable to future unseen OCT scans as well. This slide contains the visual comparison of the row target predicted and the difference between target and predicted data. We can see that our image has the predicted image is way better than the raw image in terms of clarity and is reduction in speckle noise. So the qualitative observations that we can see is that the granular speckle pattern is uh, dramatically reduced in unit outputs, and the nuclear layers that were the homogeneous regions appear more smoother and uniform. The edges are also preserved, and texture information that is also very crucial at the time of diagnosis is preserved in our model. So we also see that the characteristic foveal depression, that is the central thinning, in slice photo is accurately maintained. The peripheral performance is also good, and even in challenging regions, the unit maintains effective denoising without excessive smoothing or losing structural integrity. Um, when we compare it to baselines, unit outputs are usually smoother than median filtered images, and there is less residual speckle graininess. They appear sharper than NLM outputs as well, and the boundary smoothing uh, is also avoided in units architecture. A unit also achieves closer visual match to pseudo clean images than either baseline methods that we have used that were median filtering and non-local means, that is NLM. So we can conclude our project that uh, we have successfully reduced speckle noise in OCT images, progressing from classical image processing techniques to modern deep learning approaches. The work successfully demonstrated that supervised learning, when combined with pseudo-clean training targets, can substantially outperform the traditional denoising methods that are present. The key achievements of our projects were dataset characterization and comprehensive analysis of 250 to 260 manually annotated OCDB scans. Jitter quantification and correction was required to create pseudo clean targets, and then baseline performance establishment, which allowed us to understand the evaluation criteria and realize if we were improving uh, using the classical techniques as well. Then we moved on to supervised learnings, and then again we evaluated using PSNR and SSIM and realized that it was even better than the classical techniques. Then we can look at the clinical implications. So the enhanced visualization of retinal layers stratification facilitates diagnostic interpretation, and preserved boundaries supports accurate automated layer segmentation for quantitative thickness analysis. Which reduced bucket noise also improves detection of pathological features uh, and consistent performance across foveal and peripheral regions. Also enables application to complete volumetric scans. Um, Limitations and future scope of our project is in our dataset we only had one stack of images, volume of images from one patient. Generalization to diverse patient populations can help us to get better results. So the supervised learning was also only done through pseudo-clean targets, and the quality of those targets could have improved by understanding what the actual denoised uh, image can be, but we did not have data about that, so we had to create pseudo-clean images. Um, So beyond OCT images, the methodologies in this project, that jitter correction, via phase correlation, pseudoclean targets, and all of these things, robust median filtering, can be used and in further applications as well. The work demonstrates that deep learning, when properly applied with domain appropriate pre-processing and loss function designs, can help advance the medical image denoising beyond classical methods. The successful methodology and comprehensive evaluation framework has helped us to finally complete this project. The achieved noise reduction while preserving anatomical structures represents a meaningful step towards improving OCT image quality for both clinical diagnosis and automated quantitative analysis of retinal health. Thanks a lot.